now we get the real West Nile story. So I'm pleased to introduce Professor Uriel Kitron from the Department of Environmental Studies at Emory University. Professor Kitron obtained his BSc at Hebrew University, an MPH at University of Michigan, and his PhD from the University of California in Santa Barbara. His research and teaching programs center around ecoepidemiology of infectious diseases, with an emphasis on tropical and emerging diseases and their environmental risk factors. As we will see in his presentation today, he applies tools like these GIS tools that we've been discussing and remote sensing to gather and manage environmental data that can explain the spatial distribution of disease vectors and assess risk of transmission. He then uses these techniques in quantitative spatial analysis, mapping, and modeling to refine the research approaches and to support surveillance and control efforts by public agencies. The title of his presentation today is his work on West Nile virus in Chicago, considering the past, understanding the present, and predicting the future.
It's a real pleasure being here. And I think people that do geospatial work are kind of like real estate agents. They always think about location. So it's a triple pleasure for me. First of all, it's a beautiful building. I'm really struck by it. Second of all, the fact that this is sponsored by the library. And libraries really have become the leaders in dealing with geospatial data. And of course, we are right next to the Center for Statistics and Geospatial Data. And finally, I have a lot of friends and collaborators here in Case Western, mostly in the Center for Global Health and Disease. So it's nice to have an opportunity to meet people from other parts of Case Western. So let me say a few things about the type of diseases I work with. And they generally fall into the categories of vector-borne zoonosis. And if we talk about vector-borne zoonosis, we have to talk about the pathogen, we have to talk about the vector, and we have to talk about the reservoir host, typically a vertebrate, a mammal, or a bird. And as we have more and more new diseases, or all diseases arriving in new places, we are asking questions such as, where did the diseases come from? Where will they go to next? How did they get to a place? When did they get to a place, or when will they get to a place? When will the next West Nile virus come to Cleveland or to Chicago? We are very interested in the environment, and specifically in anthropogenic changes, in human-made changes that change the conditions that expose humans to transmission of diseases, of infection, to which they were not transmitted in the past. And then the big question is, we have invasions all the time. I think we all know that, mosquitoes, snakes, tigers, everything is being smuggled and brought into the country in many different ways. Most invasions are unsuccessful. I would say probably 99.9% are unsuccessful. We don't even know about 99% of them. The 0.01% that successfully make it, those are the ones that we are interested in, and we'd like to predict it. And I think there is general agreement that the rate of new diseases emerging or all diseases reestablishing themselves is on the increase. So if we talk about vector-borne zoonosis, we have to talk about the arthropods, which are the vectors. Of course, we have to talk about the pathogens. We have to talk about the humans and other animals. There are many vector-borne diseases that are zoonoses, and many of the emerging diseases are zoonoses. I think 2/3 of the new and emerging diseases are zoonotic diseases. Those are animal diseases that become, animal infections maybe, or animal diseases that become diseases of humans. And there are many of them. We talked about West Nile virus. We'll hear about yellow fever and about plague, I think, in the afternoon. There's a whole bunch of them, and there are probably some other ones that are coming or that we are not considering important. I used to teach the entomology part in a parasitology class in the veterinary college, and I would mention West Nile virus because I'm from Israel, and our West Nile virus came from Israel. And nobody even, it was a tiny little line. Nobody ever remembered it. And suddenly, 3 or 4 years ago, it's probably the most interesting thing for the veterinary students to hear about. So Charlie already talked about GIS in general, but specifically for vector-borne diseases. And we talk about really all the geospatial sciences. So we talk about GIS, we talk about GPS, we talk about remote sensing, spatial analysis, spatial models. As an ecologist, I'm interested in analyzing transmission dynamics. To some extent, disease I'm not as interested in, except for an outcome, and often an accidental outcome of the transmission process. I'm interested in the complex role of landscape and climate, and ideally what I would like to come up with is predictive models that consider space and risk maps that are actually effective and useful for the Cleveland or the county health department or for the CDC. The issue of scale is crucial, and it already was touched upon. We can consider diseases on many different scales, spatially or temporally. We cannot assume that what we do on one scale is also relevant to another scale. So what is happening on the county level may be completely irrelevant for the whole state of Ohio or the US, and vice versa. And we have to think both about spatial scales and temporal scales. So geographically, in Kenya or in Argentina, we study one community, and then we studied several communities. We want to then explore a district area phenomena and maybe even a national level area. Temporally, sometimes we are interested in an outbreak. There may be an enteric outbreak that we really need to track down and do something within a matter of hours or days, or may want to see how West Nile virus is changing between seasons over years, or how malaria is changing over decades in sub-Saharan Africa. And as I already said, we have to be very careful when we move from one scale to the other. We often do it, and in fact, there are often no perfect scales in which to explore this transmission of disease, but we cannot really extrapolate or interpolate freely from one to the other. So I'll talk about one example, and that is our work on West Nile virus in Chicago, an urban ecology study of the establishment and distribution of the virus and how it relates to the mosquitoes and the birds. So in order to have an active focus where transmission takes place, several things have to happen. First of all, the vector has to survive and do well. Reservoir hosts have to be present. Pathogen has to be transmitted between the vector and the reservoir host. And as far as transmission, as far as infection, as far as the virus, that's really all that matters. From our point of view, to actually have a disease focus, there have to be opportunities for exposure of humans or of animals. From the virus point of view, this is unimportant. In fact, often it's deadly. The virus often will not be transmitted farther from the humans or from the horses. We either get too sick or the viremia is too short. But from our point of view, this is what we're really interested in. And Charlie's talk was a lot about the social conditions that allow for transmission to humans and domestic animals. So West Nile virus in Illinois, it came to New York City in 1999. It came from the old world. In fact, like me, it came from Israel, because the strain in New York was identical to a strain that was found in geese in Israel a year before that. In Chicago, in 2001, we had positive bird specimens, no human cases. We had a big outbreak in 2002. We led the nation in number of cases. About 20% of all cases in the US occurred in Illinois, with 66 deaths, and most of them in the greater Chicago area. Of course, we have most of the population there as well, but there was a clear hotspot in the Chicago area. Slow years in 2003 and 2004, and two smaller outbreaks in 2005 and 2006, and again, much less activity in 2007. The disease, the infection, the virus has moved really as a wave very quickly all the way across the United States. And I think everybody agrees that it is here to stay. So if it has to do the epidemic curve, this is the 2000, can you see that work? Yeah. This is the 2002 outbreak, and these are the 2005 and 6 outbreaks, and then the very quiet years. One thing common, and I don't have much time to dwell on it, but maybe during the questions and answer over lunch, the outbreak years were very hot and very dry. I'll talk a little bit about that. So there were some unique climatic or weather conditions that allowed for transmission. These are data from the first year, And just as an aside, we talked about working with health departments. There was no real GIS unit at the Illinois Department of Public Health. So we were doing the GIS work for them. So this is actually a map that we created for them for the data on the dead birds in the greater Chicago area. And I think it always has to say, no, I'm not going to talk about, in fact, I will say as an aside about getting data and everything. Although we handle all the data for the State Health Department for at least the first 2 years, once we needed it back for analysis, it took a year of legal processing for them to allow us to use the data officially, although we had it unofficially. So that's a question that I think maybe you asked about data. Anyhow, we probably should have paid much more attention to what happened in 2001 because there were obviously two very defined foci of disease activity. One in the Evanston-Skokie area in northern Chicago, and one just south of Midway Airport in Oak Lawn and Evergreen, with downtown Chicago being around here. And indeed, what happened the next year is that the human cases occurred very much in the same foci, the two in the north and the one in the south. Those are not the most densely populated area. It's not downtown Chicago. It's not the most sparsely populated suburbs. Those are areas of intermediate human density. And I already said there are three well-defined foci. Oops. If we try to do some spatial analysis for the map, look at it within census tract or within artificially defined hexagon, it makes the clusters much clearer. But the clusters are truly and statistically significant, whether you talk about cases or you talk about incidence in disease in relation to population size. And you can do some spatial analysis. I won't talk about it too much, but this actually shows that in that case, the hexagons that have high number of cases were near other hexagons that had a high number of cases. So the apparent clustering was also statistically significant. To put a historical perspective, there's something interesting about that area, because this area of the southern focus of Oak Lawn, Evergreen, south of Midway, in 1901, the New York Times in an article talked about this area as having dirty streets, filthy alleys, impure water, the causal epidemic of typhoid fever and malaria. Very interestingly, in 1975, we had an outbreak in the US of a closely related disease, St. Louis encephalitis. In fact, in New York City in 1999, initially West Nile virus was misdiagnosed by the CDC as St. Louis encephalitis. And that focus occurred in the same area, and then we had the outbreak in 2002. So this is a summary of the data of the 75 SLE outbreak and the 2002 West Nile virus outbreak. And it's obvious that these two foci are practically identical. In fact, I think one reason we were successful in getting funding from the NSF, NIH, Ecology of Infectious Disease program, because we pointed out that we really have a natural experiment here. Something that, allowed for transmission of a very similar virus with the same vector in 75 remained valid in 2002, but something else was different. And whether that is because conditions changed or because there's difference in transmission of the virus, West Nile virus versus SLE, we don't know yet, but that's the question that we are exploring, and I think we are getting closer to an answer. So we were trying to relate the distribution of West Nile virus cases to a variety of risk factors. And much of this work was led, there are many collaborators, and I'll come to them at the end, but much of this work was led by Marilyn Ruiz, a medical geographer at the University of Illinois. So we tried to relate cases to vegetation, and like housing, the cases were concentrated in an area of intermediate amount of vegetation, intermediately green area. Very importantly, when we look at physiography, the distribution of cases was very much concentrated in the Chicago Lake Plain, in an area that is prone to flooding. In fact, this area used to be heavily flooded. The housing, it really developed greatly after the Second World War with a big housing boom. And drainage is a big problem in that area. I'll come back to that in a second. So what we did is we said, OK, we have an urban landscape. We do a lot of work with rural or sylvatic landscape. Can we define the urban landscape into different categories that will allow us to identify some set of characteristics that predispose or explain the occurrence of cases in that area or the lack of cases in another area? So what we did is we divided basically the greater Chicago area according to housing, vegetation, socioeconomic data, land use data, and came up eventually with 5 different categories ranging from new, wealthy, hilly, natural, to areas that had high density, white, low vegetation, and housing from the 40s to the 60s, or lack of that kind of housing. And what we came up with, that there was one type, urban type 5, dominated by housing from the 40s and 50s. That is the area that maybe was not developed until then because it was so prone to flooding, although there were other reasons. It's mostly white. We are not sure about that, but it does stand out as an independent variable, but we have no explanation for it. It's not only because of where the people live. Moderate vegetation and moderate population density. 64% of all the cases from 2002 were in this group. So the relative risk was highly significant, 3.5 times more cases per 10,000 people than in all other types. So this area clearly is conducive to transmission of West Nile virus, and was also conducive to transmission of St. Louis encephalitis, and was also conducive to transmission of typhoid and malaria in the beginning of the last century. So one reason, and that's all I'm going to have time to talk about the weather and climate, is the issue of drainage. The area actually is characterized by a large number of undocumented storm drains. So if you go behind the houses I just showed you, there were probably alleys between the houses, and then people gradually, I guess, moved the fence and expanded their backyards, and then they took over the alleys, and the storm drains were still there. So when we actually started looking, or when the Department of Public Health started looking, there were over 3,000 undocumented storm drains. Those are filled with grass clippings, leaf litter, etc. They hold water, organic matter, perfect habitat for Q. excipiens, the main vector of West Nile virus. That also explains why hot and dry years are very good for transmission. Hot makes sense. Everything is faster, more mosquitoes, virus development developed faster, etc. Dry seems a little bit counterintuitive, but what happens in dry years is, especially if you have a lot of good rain in the spring, the water remains standing here. The larvae never get flushed. That's why those dry, hot summers are perfect for keeping the water, organic matter, more larvae, and continuous development. If you have many rains during the summer, they get flushed repeatedly, and we have very little development. But that is, so we are pretty good at explaining the pattern. But the question is, why is this a persistent focus of our virus transmission? In that case, we're talking about West Nile virus in 2002 and SLE in 1975. And this overlap is really striking. I mean, it actually, to me, it's nearly, it's nearly too good to be true, like the Mendelian 1 to 2 to 1 ratio. But it's real. So something has definitely happened here. And to put it in ecological terms, how does the host-vector interaction determine the timing and the pattern of amplification of the virus within these hotspots? So we decided to look much more closely in the hotspot. We said, let's look very closely at what's happening in a relatively narrow area and see if we can start to understand the pattern. So that is a study that we have been doing for the last 3 years, supported by the NIH NSF program. Big collaborative group from University of Illinois, Michigan State University, people that are working from University of Torino in Italy that have visited, and many units within the University of Illinois as well, and the Illinois Public Health Department that should be here as well. So what we did is, and what we are doing, we have a big field team, and we look simultaneously every summer at the mosquito vector population at the avian host population, as well as the virus using molecular epidemiological studies. We are trying to understand, for the mosquito vectors, to understand viral amplification and who they feed on host-wise. We try to understand the avian community and the infection rate. And then we want to understand about the West Nile virus to actually look at patterns of transmission and even evolution. So we started in 2005 an extensive field study of the birds, mosquitoes, and virus within a relatively small area of about 8 by 8 kilometers. We picked 16 different sites. Some of them we determined as very urban, and some of them we determined as being more natural. Now, natural within a city is limited, but you have areas, primarily cemeteries, that actually provide a relatively natural setting. And we want to compare, among other things, what is happening in the more residential site versus what's happening in the more natural sites, and see, the interplay between those two different types of sites. So as far as the vector community, we did a lot of mosquito trapping, and you have to trap mosquitoes in many different ways. Otherwise, you get a very biased sample. So we use light trap and gravity traps and aspirators to get the adults. We try to quantify the productivity of the different larval sites, such as the catch basin. We also try to see how much the mosquitoes are producing. So we had oviposition traps, egg traps. And whenever we caught mosquitoes that were blood-fed, we did a blood meal analysis to figure out who they feed on. And without getting into too much detail, one striking thing was that the minimum infection rate, which is how you measure the proportion of the mosquito population that is infected, goes up very sharply. You look between the second half of July, within 2 weeks it peaked. That's a very sharp increase. So a lot is happening in a very narrow period of time. That is also probably the period when many birds get infected, when many nestlings get infected. We actually think it's very tight synchrony between big rise in mosquito population, big rise in mosquito infection, and the availability of naive birds explain why we have this relatively explosive outbreak in northern cities. I recently moved to Atlanta, and one of the big questions there is that we are starting to tackle, why don't we have those big outbreaks in the southern cities? What is different? And initially I thought that this curve will not be as sharp. That does not seem to be the case. It seems like it's going as high, as quickly in Atlanta as it does in Chicago. So we may need to look at the birds more closely. So as far as the bird goes, we are doing bird surveys. We have some very good ornithologists led by Jeff Braun that are leading this effort. And we are doing line transect bird surveys. We do a lot of mist netting. We have trapped thousands of birds. And when we trap the birds, we check them for seropositivity. And we try to find the virus as well. It has been very hard to find positive birds. Birds are viremic for a very short period of time. And maybe we have not targeted. We are trying now to target the nestlings. But it has been actually agonizing that we look at 2,000 birds, and we maybe have 15 that are actually viremic. We have many, many that are seropositive, but we don't know when they got seropositive. Could have been even a year ago. One interesting thing that we find out is some birds are much more important than others. And if we look at density of birds, the house sparrows and the American robins are by far the most common ones. If we look at who the mosquito is feeding on, in other words, OK, we have the blood meal. Where did the blood meal emerge from? Because then we can actually relate what's happening in the mosquitoes to what's happening in the birds and actually find not only the vectors, but who are the important hosts. And it's very easy to calculate a feeding index. Basically, you look at the proportion of blood meal from a certain species versus its density in the area. It turns out that sparrows, although they are very common, are not very important. The mosquitoes are actually avoiding them. A value less than 1 means that they prefer not to feed on. But they do like the robins, and the robins are common enough that we think they are a key player, at least during a certain part of the year, maybe the most important part of the year. Some other ones may be important as well, but we actually can say something now about the importance of a specific bird species in transmission. And then we can look more closely about the biology of the robins. I didn't go into too much detail, but one of the most interesting things we found in this study is that even within these relatively small hotspots of maybe 8 by 8 kilometers, there were hotspots within those hotspots. And we really had to look not only at natural landscape and the built environment and the climate, but also at the socio-demographic area. There was really, we term it, exceptional variation in the risk within the hotspots in all of those parameters. If you talk about the vector ecology, there was real heterogeneity in the magnitude and timing of amplification of the virus within the hotspots that we studied. If we look at the host ecology, again, the American robins that drive the amplification, the distribution and exposure was very different within the hotspots. And even if you talk about the evolution of the virus, the genetic diversity within suburban Chicago, within our study area, is incredibly high. In fact, the diversity in our study area is as high as the diversity in all of the United States. Now, we're not talking about any, directed evolution or anything like that, but as far as diversity, it is striking. And that work was led by Tony Goldberg and a student from Italy, Luigi Bertolotti, and has appeared recently in virology. So we have very high genetic diversity within the hotspots, and really what we have overall is our hotspots within hotspots. Even the relatively small urban focus in Oak Lawn, Evergreen, is not at all homogeneous. And what we really think now, if we want to go to the process, if we want to go to the ecology, we actually feel we need to go to a much finer spatial and temporal scale. We need to go less than 1 kilometer. Tony Goldberg actually would say maybe 10 meters, but certainly less than 100 meters or less. We need to look at what's happening within days, not in weeks, because that is really the driver of the transmission, the amplification, and the evolution. And once we understand those very local processes, then maybe we can try and see how do they actually coalesce to create the kind of patterns that we can see. And the GIS is so useful in pointing to us. So in a way, the GIS and spatial analysis were the drivers for us to say, OK, we have to look finer, finer, finer to actually understand the ecology. And what we hope to do in the next several years is to specifically look exactly what's happening on a very local level. So to come back to the practical arena, what we really want to do, hopefully, is explain and predict. And somebody asked, can we predict how will be the next year? And will the focus be the same? And that what we do here is relevant to what's happening in Detroit or in Cleveland, for example. And for what we have to do for that, first of all, we have to get a lot of data. And I think something that Charlie mentioned, we often don't have the baseline data. When West Nile virus came to New York, everybody kind of felt it was a big year for mosquitoes, but there were no data about mosquitoes for 20, 30 years. If we don't have the baseline data, how can we actually tell or predict what will happen? So not only remotely sensed and big spatial data, even for the kind of epidemiological and entomological data, we need to do a much better job of collecting the data before the disaster or the catastrophe strikes. So what we're trying to do is put together all this data, and we go very quickly, but the point I want to make is that one thing we do have, we have data on mosquito infection, and I'll just freeze it on one snapshot if I can. OK. So what we are trying, this is just one of the series of quick weekly transmission I showed you. We have data on mosquito infection data. We have data on West Nile virus human illness. And what we are trying to do in this case, very simply, is relate the mosquito infection rate in different locations, different times, and see will that predict what will happen as far as human cases a week or 2 weeks later. And there is a clear association, but not to the extent I can actually say, OK, the mosquito infection rate jumped way in this census tract right here. This is where we'll have the cases right away the next 2 weeks. But there is a very strong explanation. And as we use more data, as we get better bird data, hopefully we can actually do that. So what we want to do next, I already hinted to that. we think now that we really have a good understanding of the pattern on many different levels. And we are now ready to actually look at the fundamental ecological processes that drive this very fine-scale heterogeneity in the distribution of West Nile virus. And we're going to focus on very fine scale. We're going to do microgeographic studies. We're actually going to look and see if we can actually look at the dispersal of the mosquitoes. For example, what is the interaction between the natural areas and the residential areas? Is one kind of a refuge at different times? We're going to try and track daily aggregation pattern of robins. We actually would like to, basically put a recorder on them and see where they go and when they go, especially when the nestlings are leaving the nest, which we think is very crucial. And then we will continue with our molecular methods on a very fine scale. So there are a lot of people that are involved in that study, and that is generally true of those eco-epidemiological studies. You need to put a whole group of people together. And the big gap, as an aside, is generally the social scientist. We are getting better at actually realizing the gap and bringing them in, but it still remains as a major gap. So, and just to finish, so I talk about West Nile virus. I love the work on West Nile virus, but when you think about the importance, it's not a very important disease. It may sound horrible because people have gotten very ill, people have died. But when you compare it to other diseases in the tropics, but even to the potential of the next virus that may come in, maybe Rift Valley fever, this is relatively a wimpy virus as far as this is in humans. And the question is, did we really learn the lesson for the West Nile virus? Because in a way, we had a dry run for this disease that came and spread very quickly. And my answer is, I don't think we are doing as well as we could have. Because of the kind of work that Charlie is doing with the county health department, or we're doing in Chicago, some people are doing that. But all, in a way, I feel we wasted an opportunity to really educate everybody about the importance of baseline data and ongoing surveillance. Okay? Good. I'll help right here. Yeah.
Did you mention, I just want to clarify the point, that in the hotspots you identified higher genetic diversity among the viruses?
In the one area that we studied, which is about 8 by 8 kilometers, within that area, the genetic diversity was comparable to the genetic diversity reported for the whole US.
Okay. See, that's really an interesting result because it's a bit counterintuitive. You would think from population genetics, like a founder effect, a successful virus that figured out the zoonotic shift would be all of one genotype, had already gone through some selection, and then that one was taking off.
Right. Now, this is a very fine scale genetic diversity, and it's not at all directional. But the diversity is yeah, I agree.
Very interesting.
So that was actually, yeah, and interestingly, when we submit the proposal, that's the one thing that the reviewer said, that's not a very great direction to go. It turns out to be actually one of the most interesting findings. Yeah.
When you found the the maintenance schedule on the storm drains that were undocumented, how did that yield some type of interaction with the Public Works Department, something like that, or if it was a landowner's educational?
Yeah, very much so. Because, so let me say one more factor that I did not mention. There were differences depending on mosquito control districts. So at least in neighboring areas that were very similar according to the criteria we looked at, the mosquito control district actions were very important. And we've published that as well. We work very closely both with the state health department, with the county health department. Chicago is very complicated, I think maybe like in here as well, because we have the Cook County Health Department, we have the Chicago City Health Department, then every of the like Oak Lawn, Evergreen have their own people. So we worked very closely with them. And I think maybe, I don't know that for sure, but one reason that the more recent outbreaks in 2005, 2006, or 2000, were not as severe as 2002, there are many reasons. One, there could be more immunity, but one reason I think the extra control activities. And now that all the storm drains are mapped, all the catch basin, they're actually putting ultraseed tablets, which are insect growth regulators, which prevent the larvae from emerging as adults. And I think that has a lot of impact. So yeah.
I don't know if I have my diseases mixed up, but is this the illness that was killing birds? Was killing crows and blue jays or whatever. Was that part of uncovering what was going on, or is that part of the dynamic?
Yeah, it's actually it's kind of unusual because the classical zoonosis will not impact the wildlife. If you look at the old world, in Israel, in Africa, birds are not impacted. Even corvids, even closely related birds to the crows and the blue jays, it was so heavily impacted. So this is the unique situations where a virus came to a completely naive population and had a major impact on the bird population that probably were never exposed to anything like it. As the disease is spreading south to Mexico and farther south into Americas, it seems to be a little bit more similar to the old world. It doesn't seem to have the impact on the bird population. So, but yeah, in fact, for a while people are actually literally talking about birds falling from the sky in New York in '99 and in Chicago in 2001. So it had a major impact, and the population has not recovered yet. There are still fewer crows in big parts of Chicago. That's a whole interesting question. How will they recover? Will they now be, have a certain degree of immunity? Those are very interesting questions from a wildlife conservation point of view. Yeah. Pete?
So the two talks have shown very nicely how adding more detail help us understand the mechanisms at the ecological level of disease transmission. Let's say that we were dealing with a pathogen that came into the United States and mortality rates were 5-fold higher and were really causing alarm. Can we actually use what we've learned from this kind of gentle reminder that we're exposed to these viruses? How quickly can we respond in this country to something that was a more serious public health outbreak? And what would be the things that you would suggest we equip our cities with in order to prepare ourselves for that kind of more important outbreak?
Okay, could people hear the question? Yeah, okay. So to me, that is actually the key questions. And in Atlanta, we just actually last week, we met with people from CDC and University of Georgia and with a Georgia State entomologist. And she has been really a leader in studying what's happening with West Nile virus in Atlanta. She told me that basically her budget, and Atlanta is suffering from major budget constraints, or Georgia is, but it means Atlanta, her budget has been completely cut. She has nothing. They were doing some surveillance, they had the, the light traps, they had the mosquito collection, there's nothing anymore. So whatever is being done is done because some people at CDC, like Tom Burkert, or people at University of Georgia, or now ourselves in Emory, are actually saying, we actually need to do the kind of research that will also provide the surveillance data. And in fact, unlike, well, I think in most areas, even when they did surveillance, it was very much like the, the drunk looking for the coin under the light, because they were studying where cases occurred. So that's very important, but it's equally important what's happening where there were no cases. When everything else was similar and we didn't have any transmission, we need to study that. So I think it's coming back to surveillance and to funding the very non-glamorous collection of baseline data and having a continuous database. Because then, if you talk about, let's say, a more extreme virus, several things would be valuable. One would be an early warning. Even a few days can make a big difference, especially for this disease that have a vaccine. But either way, there may be reasons why we can do something very quickly. And the other one is, even under ideal scenarios, we're also always going to be working with very limited resources. So anything that will help us target our limited resources, be it drugs or larvicides or people or anything, will be very useful. And I think the kind of work that Charlie has been doing here, that we are doing, and other people are doing in other places in Louisiana and elsewhere, is relevant to that. Because at least I think, we, for example, can say, okay, let's say Rift Valley fever. It's a good example because it's a much more serious virus, and the vector is the same as the one for West Nile virus, generally. Well, then maybe we can say, okay, if we know that there are some cases of Rift Valley fever somewhere in the US, we can maybe predict which parts of the big cities are at risk. And this is also a livestock threat. So we can actually say, here where we should target all our efforts. So I think to that extent, we have this data, but it's getting lost as things are changing.
So if there's a boring levy on your ballot, and it's a public works levy, you would encourage people.
Oh, yeah, right. Yeah. And it's very hard, because whenever, like even West Nile virus, now there is much less of it. I presume you experience the same thing in the county, that the support is kind of dwindling as you have fewer cases? So.
Yeah, I'd say one of the big aha moments that we had, we did everything we could to take the political boundaries out of the equation because it wasn't natural, it was all artificial. But then you realize that politics drives the appropriation for mosquito control, the sensing efforts, and so on. So then you had to go back and readjust for what city you lived in, what district you lived in. And that turned out to be important.
I think we have time for one more question, if there is one.
Thank you very much.